Okay, so yeah, today I'm going to be talking about the role of functional and structural brain imaging in educational neuroscience. And we'll see that there's some kind of irony in that educational neuroscience itself was kind of launched or propelled forward by innovations in functional brain imaging. So then it's kind of ironic about to even ask the question about how's it going to be useful in the practice of the field. But that's what we're going to get into today. So here's what I'll talk about. For those of you who don't know about educational neuroscience, I'll give a very brief overview. Now, terminology-wise, in the US, this field is often called mind, brain, and education, but it does go under other names. Sometimes it's called neuroeducation. Sometimes it's called even neurodidactics. I'm not sure we have the best name for the field yet. I don't really like educational neuroscience. In fact, maybe halfway through the talk, maybe I should just change the name. The problem with educational neuroscience is it makes out that neuroscience is to the fore, and this is just a branch of neuroscience. Whereas really the whole enterprise is to bring together people with different disciplinary expertise with the common goal of improving educational outcomes. Okay, so then I'm going to talk a little bit about how brain imaging is received within the field, and particularly amongst educators and teachers. And then we'll get into the main part of the talk where I will consider 6 different possible roles that brain imaging can play in educational neuroscience. The ones at the end I might go into a little bit more depth, but I will try and give these different possible roles marks, and then we can look at our score sheets when we get to the end. Okay, so let's start off defining educational neuroscience. So as we've heard about, the goal of this field, it's intrinsically transdisciplinary. The goal is translating insights into neural mechanisms of learning into education to try and improve educational practices, ultimately learning outcomes. And there has been a very long history in modern psychology with interaction with education. The original Stanford Binet IQ tests back at the beginning of the 20th century were all wrapped up in optimizing educational outcomes. But educational neuroscience is, as I say, driven by new developments in brain imaging, really in the 1990s, but also in understanding how learning works and different ways that it can work formalized in machine learning. So together, these fields came together with to generate a new science of learning. And as I say, part of this was driven by the decade of the brain, as it was christened in the US in the 1990s, which had the goal of enhancing public awareness of the benefits to be derived from brain research. So as the introduction said, I'm the director of the Center for Educational Neuroscience. That is a cross-university research center that was set up in 2008 to bring together expertise from University College London, the Institute of Education, and Birkbeck College. So people have different expertise, but broadly, UCL has a strong expertise in neuroscience. Institute of Education, self-explanatory. And in Birkbeck, we have a very strong focus in developmental cognitive neuroscience. So we're about a dozen faculty members who work together on different projects with this goal of translation. Take basic science and see how that might be useful in an educational context, but also to listen to educators and use their interests and priorities to try and direct some of our research. So let's move on to how educators and education researchers view brain imaging. Not always welcomed with open arms. So it's coming out, what, 5 years ago now, we published a paper which is sort of giving an overview of the field and where it's at. Still a relatively young field. So it has some growing pains, and there are some, I think, legitimate criticisms that we try to address as we go along. But there are also some distractions. Amongst the criticisms, you will hear that the kind of thing that neuroscience looks at, the kind of data it generates, is just too level to be relevant for education. So as they say, there's no direct path from brain scan to lesson plan. Activation of brain areas, how is that going to be useful for a teacher in a classroom? And that is a big challenge to think about the number of steps of the translation process and how important it is to involve teachers, educators in negotiating that translation process. Often more an art shaping a dialogue than a science. Because of that, some reservations that as it started off, the field felt like neuroscientists, cognitive neuroscientists felt like they had an understanding of learning mechanisms, and it was their job to tell teachers about that. So a bit kind of preachy, and teachers might legitimately feel that these neuroscientists should stay in the lab. And what did they know about teaching a room full of 36-year-olds, which is kind of fair comment. So a lot of what we do in the field is to think about how we build the dialogues between educators, researchers, both within psychology and neuroscience, and at a higher level, policymakers as well. And then also doubts just about the viability of neuroscience methods for sort of diagnosing disorders or studying individual differences, just whether in practice these methods are sensitive enough or economically sensible, whether they're cheap enough to be used in practice. And that's slightly a moving target because these methods get more portable, get cheaper, get better sensitivity, and so forth. So those are all things that the field is moving forward, and we try and work on those areas. There are some distractions though that kind of wind me up. Depends on the day that you find me. So some of them are kind of neuromyths. There's a whole area of research about how teachers and the general public misunderstand neuroscience, and it's kind of turned into a research field, which is kind of odd because that's a public communication of science issue. It's not the translation of research findings. I mean, it's sort of tangential, and I resisted for a long time doing research around neuromyths. In the end, we ended up putting a resource on our website to debunk various neuromyths. But the big challenge is if you go around telling people that everything they thought they knew about the brain was wrong, you do really have to have some message to replace it with. So how does it work in a way that's relevant for teachers? Another distraction, something called the seductive allure of neuroscience. The idea that somehow neuroscience is a kind of framing effect, that if you suggest some new teaching method, but you illustrate it with brain images, people are much more likely to believe what you're talking about. So it's kind of all smoke and mirrors. I would say both neuromyths research and seductive allure of neuroscience research are kind of subtle ways for education researchers to undermine or to criticize educational neuroscience. And then, of course, we have commercial providers who want to, sort of as endorsed by neuroscience or neuroscientists, to push commercial products, which may have very little scientific evidence supporting them. So a couple of examples of papers is a systematic review. There's enough literature on neuromyths out there to now do systematic reviews of neuromyths research in educational settings. And here's the original paper on the seductive allure of neuroscience explanations. So I think you do have to be careful and thoughtful about thinking about the whole piece, the whole context of education, and where neuroscience fits into that, and not make over strong claims about the relevance of even just learning mechanisms. Learning mechanisms are only a small part of education. They are individual level factors. And what I'm showing you here is a diagram, which is kind of a mashup of Bronfenbrenner's ecological systems theory, but also Susan Mitchell's behavioral change wheel, just showing the nested constraints on behavioral change, in this case on educational outcomes. So in the middle, we have the learning outcomes, but we have these individual level factors to do with motivation, attention, nutrition, ability, health. That's probably what our understanding of brain mechanisms can inform. But what the individual does and what they can achieve is a little bit constrained by the educational environment, the school policy, classroom environment, the teacher skill, teaching materials. And then there are wider social influences around that. So the family, socioeconomic status, cultural influences, cultural expectations around learning, availability of technology in homes and kids on computers and mobile phones. And then we have the broader sort of national context of governments determining what should be taught and what not taught, and the educational budget and how you structure your education system, and train teachers and employ teachers, and education policy and so forth. So one can't say neuroscience is everything. One just has to understand that the context within which it works. And then how is educational neuroscience going to work? What is the pathway between education research and neuroscience? Some people would argue that the link is always indirect, that neuroscience is telling you how to improve your psychological theories, how to have better theories of how executive function works, or literacy development, or the role of emotions on attention, or special causes of learning deficits in special educational needs, and so forth. But then there is a possibility, I think, of direct links between neuroscience and education. And that's when you think of the brain as a biological organ with certain kind of metabolic needs. And then you're looking at things like the role of aerobic fitness, the role of diet and nutrition, of when you eat, when you don't want to turn up to school hungry, the role of stress, the role of sleep, and so forth. So that's really about putting the individual brain in the classroom in the best state to learn. And you don't necessarily have to appeal to psychological constructs to think about ways to optimize that. And I do think it's important that we recognize as sort of basic scientists or cognitive neuroscientists the limit of our expertise. So teachers really are the experts in the classroom. Yes, researchers may know about things like cognitive control and working memory and language and planning and the relationship between those, but that is very different to getting a class of 36-year-olds changed into their sports kit, or to pay attention in maths class and so forth. And I think a lot of the time, we're not trying to come up with miraculous silver bullets, things that teachers just haven't thought of doing, that come out of the neuroscience that will revolutionize education. A lot of our bread and butter work is going to be looking at the things that teachers do that seem effective and understanding how those work. And if we understand how something works, we're in a position to make suggestions about optimization. How can it work a little bit better? And if you go into that classroom with the 36-year-olds in there, it's kind of chaos, and like kids are running around the whole time and messing around and playing and putting out materials and taking in materials and collecting up work and queuing up and making interludes where they have some songs and stuff like that. And that may be like maths class, maybe like that. So it may well be that the majority, maybe even the vast majority of time in the classroom is not active, engaged learning. But I think that just places all the more emphasis of that 10% where they are actively engaged. You want to do that in the most efficient, optimized way possible. And that's where an understanding of learning mechanisms and the cognitive constraints that maximize the operation of those learning mechanisms can come into play. So I'm showing you here a slide. This is routinely what I would show to teachers if I'm talking about educational neuroscience. So you don't have to believe this stuff, but I would say, remember, don't be impressed just by brain images on their own. I mean, you know, they're nice to look at and everything, but it's you need messages about how to do things in the classroom in a way that may be better for producing good learning outcomes or good attention and so forth. Don't waste money on brain training applications. Don't believe in neuromyths. Okay, so those are the kind of caveats that I need to put out to educators when I talk about educational neuroscience. Okay, so now we're moving on to possible roles of imaging in educational neuroscience. And we'll just start with the idea that maybe you just want to use your cognitive neuroscience to understand more about how education relevant abilities work. So, for example, here's a couple of papers. There was a paper I was reading the other day that I was quite fond of that demonstrated that you couldn't find language areas in the brain that were sensitive to syntactic violations that weren't also sensitive to semantic violations in sentences. And there was an argument here that there are no syntax-specific areas in the brain, which is going to annoy some kind of Chomskyians, right? And that's kind of interest. And maybe that's going to tell us something about the patterns of developmental language deficits we might expect, that we probably won't expect anything that's syntax specific. But does that have an application directly in the classroom? Well, not really. We're just kind of finding out about how language works in the brain, right? On the right-hand side of the screen, I put up a slide that I include in a lecture I do on developmental cognitive neuroscience of dyslexia. And that's a paper looking at language effects in the brain. So how the brain lights up differently when you're reading Chinese versus English, compared to differences between typically developing readers versus dyslexic readers. And the main point of that paper is to say you can find language differences, slightly different regions being activated in Chinese versus English, and you can find dyslexic differences, but they are different. So dyslexia is on a different axis from different language effects, right? And again, that's interesting. Does it have a direct implication for the classroom for how you would teach a dyslexic child? Well, not really. I mean, these are just kind of basic findings about how things work. And you can do this kind of work, say, in how executive functions work in the dorsolateral prefrontal cortex, or what's the approximate number system in numeracy and the interparietal sulcus and so forth. But basically what you're doing is figuring out how things work. And it is true that it's an option to keep that cognitive neuroscience research as being non-translational. For me, that seems like a bit of a wasted opportunity to be doing all of this work around understanding how education relevant abilities develop and how they work, and then not to try and do some kind of translation with that. Because if the researchers don't do it, it's not like the educators can do that translation. They cannot go reading those functional brain imaging papers and draw the lessons from them. But if you're super cautious and conservative, you might just argue that there's nothing ready here for translation. We just don't know enough to make strong claims. That was an argument made by John Brewer in the 1990s when some of the early work on synaptogenesis and sensitive periods in development was co-opted to support the sort of Clinton administration focus on early years education. And Brewer argued that that's a bridge too far. We just don't know enough about the basic neuroscience to make claims that are going to be relevant to the classroom. If you work in educational neuroscience, you seem to hear an awful lot about bridges, bridge too far, multiple bridges, building bridges between disciplines and so forth. I even published a commentary a few years back saying, enough with bridges, had enough of bridges. Let's just do interdisciplinary research, leave the bridges out of it. Okay, so we've had that first, the kind of basic science approach. The next approach is to think maybe we can use our brain imaging to contribute diagnostic tools for neurodevelopmental conditions or to look at individual variability. So there's a Stanford News web page there. Brain scans can predict children's reading abilities. Stanford researchers say that's back from 2012. And they were tracing, I think, white matter structure in a longitudinal study and looking how it seemed to be in step with the children's developing reading ability. And there's another bigger paper there on the right looking at measurements of cortical and subcortical brain morphometry, comparing individuals with autistic spectrum condition and typically developing controls. So you can find these differences in structure, but I'm not really convinced that you can use this as a diagnostic tool. And that's because once you move into an area of diagnosis, the bar gets much higher. You really need to do any kind of screening, high sensitivity and high specificity. It's got to pick up the cases and it's got to not have false positives, right? And anytime you miss individuals or overdiagnose, there are consequences. And there's research around how screening programs work and how you can, you only want to do screening if you can pick up all the cases, not overdiagnose, and there's an available intervention which is economic to roll out following your screening program. So there's a quote there I put from sort of medical analysis of screening programs. They say all screening programs do harm. Some do good as well. And of these, some do more harm than good, more good than harm at a reasonable cost. So that's the sphere you are going to need to move into if you think your brain imaging technique is good for diagnosis. Myself personally, I don't think when you look at behaviorally defined developmental disorders like ADHD or autism or dyslexia, conduct disorder, I don't really think you're going to see major differences in brain structure. I don't think that these kind of behavioral differences are reflected in macro measures of structure. If you get big enough samples and you put your machine learning on them, I'm sure you can come up with minor differences. But I don't think there's some evidence, for example, that in ADHD there's overall slight differences in overall brain size and stuff, but it's just these are small effects. Most of the differences are going to lie in functional differences. And as soon as you get into functional imaging, then you have a problem about what is cause and effect. Are you seeing, say, less activation in superior temporal areas during reading because an individual is dyslexic, or is that the cause of the dyslexia? It's very hard to figure that out without large-scale longitudinal studies. They have to be large-scale because behavioral developmental disorders are heterogeneous and there's often comorbidity. And large-scale longitudinal functional brain imaging studies are exactly the type of studies that you tend not to see. You can find kind of macro structural differences if you look in genetic syndromes like Down syndrome, Williams syndrome, and Fragile X. But then all sorts of things are different in those genetic syndromes. I'm doing some work at the moment looking at neonatal structural brain imaging for infants and babies with Down syndrome and seeing whether brain structure predicts cognitive abilities at the end of the first year. And it doesn't really work, and it's quite hard to do because as soon as you have global differences and big regional differences in brain structure, some of your registration template matching algorithms just stop working, and then you have to go back doing everything by hand, and it all gets very difficult. Okay, so general skepticism there about whether you can use brain imaging techniques for diagnosis in education. An area, though, you may be able to use this kind of research is a little bit more at the basic side, and this is to use imaging to feed into a better understanding that you don't really have discrete categories of developmental disorders, that really these are dimensions of variability, And perhaps the brain imaging evidence is a convergent way to understand what those dimensions are. So I put up a paper by Duncan Astor's team in Cambridge, where they recruited a big bunch of, there we go, 479 children who have been referred to the study on the basis of learning-related cognitive problems. So these were kids recruited because they were struggling to learn at school. And after that, the researchers said, don't tell us anymore. We don't want to know about diagnosis. We're just going to do a big battery of tests, and we're going to do some measurement of structural brain regions, regional structures, and we'll do some DTI work looking at connectomes. And then we'll just let our machine learning loose to see whether you can pull out any sort of clusters or latent categories of kids with different profiles in there, and whether that lines up to any clusters you can see on the regional brain structures or in the DTI connectome. And these researchers found that there were some profiles, some that are a little bit more language impaired and some which are a little bit more non-verbal impaired. Particularly, there's a difference between kids with executive function issues and other kids with phonological problems. But those didn't really line up with the diagnostic labels the kids have got. The profiles they found in behavior didn't really line up with the kind of similar clusters they pulled out of the regional MRI. It did line up a little bit more with the DTI, the connectomes, suggesting that maybe underlying sort of cognitive profiles line up more with connectivity than the size of different regions. So maybe that's a way that the imaging can feed into research about developmental conditions. This is one of my favorites, using brain imaging to test cognitive models. So I'll just give one example of a paper here because I was a co-author on it. So this is looking at reading, and this is a kind of an old model now, originally an early artificial neural network model looking at the reading system and arguing. So if you look at this diagram, you'll see the idea that reading is going to be a mapping of orthography to phonology, but also you might want to map from orthography directly to meaning, and there's a mapping between phonology and meaning. And then context will play a role, no doubt. But what's important here is, apart from the multiple pathways, lots of bidirectional connections. So claims for multiple pathways and interactivity. So can we test whether that is actually how things operate in the brain? So this is a paper we published, boy, when was this, 2011 now, where we were trying to use dynamic causal modeling as a direct test of this model. So the idea, I think we had about 20 young adults, and we were using multiple tasks with them. So we were getting them to read written sentences or sentences with scrambled words, or doing that in the auditory modality. And we had like baselines of false fonts and nonsense words and words played backwards and so forth. And we used this approach to try and identify in each individual what the key regions were for either unimodal representations of phonology or visual word forms, or amodal representations of word meanings. And then we built a dynamic causal model to try and understand that as individuals read, what are the changes in the functional connectivity over time between these different regions, to try and work out what regions are driving changes in the activation in other regions. So this was using a family analysis approach and Bayesian, geez, I wrote it down here, BMA, Bayesian model averaging, where we considered lots of possible relationships between these different regions that would explain the time series change in the bold signal. And what you ended up with in this model was a demonstration that, first of all, that the visual input, so this is occipital temporal area at the back, or more occipital, sort of visual input area, that direct from that visual input, you could get driving of phonological areas in posterior, superior temporal areas. And they didn't have to go via what's sometimes called the visual word form area. Yes, you could get then driving of visual input of the visual word form area, but you seem to have bidirectional relationships between superior, posterior temporal and occipital temporal areas. And then both of those areas could feed semantics. So really, this was a validation that there are multiple pathways between visual input and representations of meaning in the sort of anterior, posterior temporal areas. Sorry, anterior, superior temporal areas, but also bidirectional activity. The pathways, at least 3 different pathways could be followed to go from print to meaning. So I'm a big fan of trying to use functional imaging to test causal models, to test cognitive level models. But of course, you have to turn up with a model like that in hand to use the imaging data in that way. So another possible role is in construct validity. Now, for those of you not familiar with your validities, so construct validity, one of several validities. Construct validity is about how well a test measures the concept it was designed to evaluate. And I'm going to suggest that brain imaging can be useful for convergent validity. So that's the extent to which your measure corresponds to measures of related constructs. So this is a paper we had published a few years ago now. And this was looking at intelligence. We had a longitudinal study following teenagers where we looked at their intelligence across the whole battery, age 13, and then came back and looked at them when they were 17. And particularly, we were looking at verbal versus non-verbal intelligence. Now, previously, the general view was that intelligence stabilizes around age 10. Obviously, that's of educational relevance if you want to do streaming and setting and so forth. And we demonstrated in our data that you could actually get quite big changes in verbal and non-verbal IQ across the teenage years. So we're talking about changes in IQ of up to 20 points, either in verbal or non-verbal, over that 4-year period. Now, if you're an educational psychologist, you say, well, you're using different IQ tests at different ages, and it's probably just measurement error. I wouldn't bother about that. That's just noise, right? But what we did was structural imaging at the same time, and we were able to show that the changes in verbal intelligence were correlated with structural changes in gray matter density, in left hemisphere motor, language motor areas, and that the nonverbal changes were correlated with changes in gray matter density in an anterior cerebellar area that is involved in finger movements. So the point there is not particularly making claims about that nonverbal intelligence involves fingers or anything like that. The point is you're using your brain imaging data to validate that the behavioral data is real, that it's not measurement error. If the changes in intelligence across the teenage years were measurement error, you would have no reason to expect correlated changes in brain structure. So this is just a way of using brain imaging as convergent evidence to be more confident about what you're measuring. So now we'll move on to the kind of meat. Really, this is education. That's about learning. It's about training effects, right? So this is really where surely brain imaging is going to come into its own. So let's have a look at that. We know some research here, so the old literature, you can find that London taxi drivers who have the knowledge, know about all the roads, they have bigger hippocampal volumes. Obviously, we can't be sure because that wasn't random assignment to condition that having a fat hippocampus drives you to want to be a taxi driver. There are other studies that looked at intensive practice at learning to juggle and finding changes in sensory motor cortex. They were temporary, went away. There was a paper I quite like looking at piano practice and age effects. And amongst expert piano players, asking them to fill in questionnaires about how much practice did they do at different ages, and then using that questionnaire result to correlate differences in white matter, and finding that if you practiced massively in childhood, you could find differences in more low-level sensory areas, in white matter tracts. But if you did loads of practice as an adolescent or an adult, then you found the white matter differences in slightly higher, more cognitive type areas. So it's interesting about different areas of the brain, maybe plastic at different ages. In terms of function, we do know that with training, as you get better on something, that there is reducing involvement of prefrontal cortex with training, and a shift from frontal basal ganglia loops to motor basal ganglia loops with automaticity, sort of front to back shift. So we'll have a little look at that. Here's some, so lots of quick sum. 5 plus 6 equals, yes, 11. 8 minus 5, 3. Just lots of sums, addition and subtraction with numbers 1 to 10. And if you look at over age, kids get more accurate at that. So they reach ceiling pretty quickly. If you look at their speed, they then continue to get faster through the teenage years of doing that. This is work by Rivera et al. If you then look at the functional brain activation over development, you find decreasing involvement in prefrontal areas as they get better on the task, and increasing involvement in the kind of posterior parietal and temporal areas, as if you are sort of automating and pushing back to the conceptual, procedural, fact retrieval areas, and you are requiring less concentration and executive function involvement in order to do these tasks. And here's a task actually tracking in an individual. This is an adult who was an adult illiterate who was being taught to learn to read. And you can see, you show that individual written sentences, and when they can't read them, you just see some confused visual activation in occipital cortex. Some hundreds of hours later, after they have learned to read and got some functional literacy, now you can see all the expected visual word form areas and oral language system areas being lit up like a Christmas tree. So we can see these kind of changes through learning. To say there is a slight problem with learning, and this is kind of intrinsic to educational neuroscience, that what is one thing in psychology or education is frequently many things in the brain. So for the educator, learning is the process by which students acquire new skills and knowledge, and they can then do new activities out in the world. For the neuroscientist, learning is about behavioral change supported by changes in connectivity between neurons in potentially multiple systems. So I'm going to show you a really messy slide next. Apologies, but that's, neuroscience is messy. So these are all the systems involved in learning. You've got sensory motor cortex in conceptual learning. You've got your prefrontal cortex in control and decision making. You've got your basal ganglia loops and cerebellum in procedural learning. You've got your limbic structures involved in emotional learning. You've got the conditioning system, so your nucleus basalis is ready to get some information from the amygdala and light up the cortex with acetylcholine. You've got your reward learning system in basal ganglia and ventral striatum. You've got your episodic learning system in hippocampus and your medial temporal lobe structures. And then, a big thing in learning is how much effort do you want to put in? And that's it's been calculated by the brain by an interaction between basal ganglia, anterior cingulate, ventromedial, prefrontal cortex. And this whole system, so like the language brain is using instructions to figure out how to do things, and the social brain is using observational learning to figure out how to do things. And all of those systems have different rates of learning and consolidation and forgetting. So, I forget what the capital of Peru is fairly easily, but not that I'm afraid of spiders. If I want to learn concepts, it's quite good to interleave different topics. If I want to learn a procedural skill like learning the tango, really bad idea to interleave that with learning the foxtrot. I just get completely confused. And then if I'm an adult, I want to strategically use previous knowledge rather than learn sensory motor primitives from scratch. So that's learning in the brain. It's a lot of stuff that's going to make functional brain imaging a little bit challenging. Okay, so I need to speed up a little bit here. So I'm going to talk about a project that we've been involved in, which is almost coming up for a decade now of working on this project. This is really trying to do a proper educational trial to test an intervention and use brain imaging as a part of that. Now, if you're going to do that, it's going to take a long time. There are a lot of steps you have to go through. You've got to have a feasibility study, a mechanistic study, an efficacy study, efficiency study, and then actually turn it into some kind of activity that teachers want to use. And we are, at the moment, on this particular endeavor, we are up to an efficiency study. So a quick quiz. You should all be good at this. Shouldn't be too threatening. Does the sun go around the earth, or does the earth go around the sun? Well, we all know the earth goes around the sun, even though it's quite hard when you see the sun dropping below the horizon to remind yourself that it's actually the horizon rising up to cover the sun. But there you go. We can get our heads around that. Is the earth flat or round? Well, it really looks flat. And if you're 6 years of age, you spent quite a long time playing football on pitches that feel flat. And then the teacher tells you, no, the earth is actually round. Fractions, kids have learned now, they're all very good to know that 3 and 4 is 7, but is a third and a quarter a seventh? No, it's not. It's 4.083 sevenths. So somehow we have to be able to ignore the previous knowledge there that we have from learning numbers when we go to fractions. Is a dolphin a fish? No, it's not, but it looks like a fish. You find it in fishy places. It does fishy things. Somehow we have to learn that a dolphin is not a fish. Again, a bunch of, is -5 bigger than -1? So 5 is bigger than 1, but -5 is not bigger than -1. And falling objects, again, you guys are highly educated. You know that if there's no air resistance, the feather and the hammer fall at the same speed. Although probably you've never seen this, you just learned it from science. If you go on YouTube, you can actually see this is an Apollo astronaut dropping a feather and a hammer. And yes, they do actually land at the same time. So all of that is a classroom challenge that children struggle to learn counterintuitive concepts. And we think that when you learn new concepts, you don't overwrite previous concepts, but you learn them alongside the old concepts, and you use inhibitory control to inhibit the sort of intuitive or irrelevant properties of the previous knowledge. So we have some neuroscience theory about what's going on here, and that suggests a possibility that if we trained inhibitory control skills and we embedded it in science and maths content, maybe we could improve children's understanding of counterintuitive concepts. And again, we can do some basic research around this. This is done with teenagers, getting them to reason about intuitive and counterintuitive problems in maths and science, like perimeter and area and stacking of molecules and so forth. And we can actually look at the areas that are more activated in their brain when they do that, and see that they do happen to overlap with inhibitory control areas in dorsolateral prefrontal cortex. We've got a theory about what's going on. We have a classroom challenge. Now we need to try and turn that into a learning activity that children can actually use. We've had a lot of working with experts in AI and education, and working with teachers to come up with a kind of game show game where kids would be asked questions. So what's the question? Here's your question. 3 daisy plants are given different amounts of water. Click on the plant that will be healthy after 2 weeks when all other conditions are right. So that's an intuitive one. And then what about playing the music? Is that going to work? And so forth. And we have explanations of the candidates about why you would pick various options. And we then do some pilot studies and demonstrate that training inhibitory control, where you ask kids to stop and think, before they give their response actually helps them access the counterintuitive answer to the question. Demonstrate it works in a pilot study, albeit that was like several hundred children. Teachers really enthusiastic about that. Wonderful. That's helping my kids. Now you have to do a big trial. This was with 6,000 children in the UK to demonstrate it actually works under ideal conditions. And indeed, this was run by the Education Endowment Foundation and the Wellcome Trust in the UK as a proper randomized control task. Effect size not huge. You don't get huge effects in these trials, about 0.1. And now it's at the efficiency study stage, so an even bigger trial. Now, look, when we were doing this, we're the scientists, we want to check that it works how we think it works. We want to look at mechanisms. So the stop and think, that's our inhibitory control training approach. We had an active control because we wanted to check it wasn't just being in an intervention and playing on a computer that did the job. And we had a teaching as usual baseline. Nested within that, we had some kids, and you can see the participant numbers here, do behavioral tasks as well around inhibitory control and reasoning and so forth. And nested within that, we had some kids who we did brain imaging, functional brain imaging, time 1 and time 2, before and after the intervention, to have a look at training effects. So I won't go through the numbers other than you can see from this study that by the time you've got kids back for a follow-up brain imaging study across this kind of design, you've not got many kids left, despite our best intention. We were looking at 8-year-olds and 10-year-olds, not necessarily easy to scan kids in this age bracket. Okay, so we found some stuff that, you know, the areas that of the brain that are involved in doing the science tasks and doing the maths tasks. And you can see change in this group over 8 months, more activation in posterior cortex as you'd expect. But now we look at the training effects. This is in a training task that we know in a large RCT has an effect on actually far transfer to end of year maths and science tests. I think you can see from that, no effect whatsoever. So that just tells you that despite our best attempts to engage, use functional imaging to look at intervention mechanisms, the method is just not really practical or sensitive enough to help us here. All right, I'm going to jump through that. It did tell us more about what's going on in the brain during these tasks, but not the intervention component. So when we get to evaluating interventions and training, yes, very large doses of training and environmental exposure can show up in structure. It's not clear what that means for closing gaps in educational outcomes, for example, because we don't necessarily know the mechanisms. When we use functional imaging, often we're not really seeing the consequences of that showing up. It's not big enough effects. Okay, final possibility, which I'll just take 2 minutes on. Maybe you want to use brain imaging for policy impact. Okay, so here's a kind of very important paper published by Kim Noble's group, the pediatric neuroimaging group back in 2015, showing measurable effects of differences in socioeconomic status on brain structure in children. So small effects, 1 to 2% of the variability, but cortical surface area correlating with family income and with parental education. And indeed, in regions which seem to be processing language or executive functions, which line up with the behavioral differences we see, which correlate with socioeconomic status. These kind of data make it feel like, because it's kind of language-based and executive function, that it's maybe the causal explanation of differences in cognitive stimulation. There are other data which are looking even earlier and finding differences in gray matter volume in 1-month-olds that correlate with SES. And that doesn't look like it's cognitive stimulation. That makes it look like it's differences in prenatal brain structure and early brain development. So maybe there's more than one thing going on. And there's functional work. showing sort of differences in this is in selective attention. Need to be a little bit careful in this area to not have some kind of normative framework about SES, and to be open to the possibility that these are adaptations of children being raised in different environments. So you can do this kind of research, and it has big policy relevance, potentially for education, say about poverty and life chances. But sooner or later, if you work in translation areas, you are going to find yourself in front of some important policymakers who are going to ask you tough questions about what that means. For example, here's me in the UK Parliament being asked by a select committee, can I ask specifically, start with Professor Thomas, I can ask you, you could explain what is the practical policy relevance of the work being done in neuroscience and how that influences the understanding of life chances. The problem is, what do those brain differences mean for the life chances of children coming from poor backgrounds? Now, it's good that policymakers are interested in that, but it's tough to answer that kind of question. And that's because we don't know mechanism. We don't know what it is that's causing differences in brain structure. So this is an example of a paper I had earlier out this year, trying to use artificial neural network models to figure out what is the relationship between brain structure, behavioral development, and possible pathways that socioeconomic status might work via, what kind of influences in cognitive stimulation or early brain development. And it's tricky, right? Because you need to, SES is an individual differences factor, but you need to embed that in development, in change with age. And also many of the differences between individuals are genetic in origin. So your model has to have genetic and environmental factors in. So I won't go into detail on that, other than you need a mechanistic understanding to say what the consequences are. How could things be different? And it's advantageous with models that you can try interventions on them and see what the consequences are for interventions. For example, if the causes of SES differences are due to differences in cognitive stimulation versus early brain development. Okay, so we'll wrap up. Ironically, progress in neuroimaging effectively launched educational neuroscience, but either the reception from educators can be frosty, or it's too seductive, it's too reductionist, too deterministic, or over-enthusiastic, and as you get all these kind of neuromyths. Even if you're getting insights into learning mechanisms, you have to accept that actually learning mechanisms are a relatively small part of education as a field. Imaging probably insufficiently sensitive or practical for diagnosis. Possibly not really sensitive enough for training effects either. For me, the best role is really to tell you how things work, and that to give you clues about how that can affect practical approaches to teaching. I think brain imaging evidence can be persuasive for policymakers, but it's a double-edged sword, right? Because the risk is that if policymakers see that poor kids have smaller, thinner brains, maybe they'll think that that's inevitable and deterministic and nothing can be done about it. Everything I've said about brain imaging can also be said about the involvement of genetics in education, another kind of live topic. That's another story, but very similar kind of concerns in that area. So that's my score sheet. Some things you can use brain imaging for, but I don't think all of them. Best contribution is how it works. And just to say many thanks for your attention, and thanks to all my collaborators and funders.